how are you? Today this is the continuity of the previous lectures, and in the previous lectures we discussed the concepts of phylogenetics and different types of phylogenetic trees, and then we go into the details of how those different phylogenetic trees work. And first we talked about character-based methods, and then we talked about different types of distance-based methods and how to calculate the distance, evolutionary distance between the two sequences or the set of species. And after that, now we are going to discuss an example of distance-based method. That's the most simplest example. That is the UPGMA method, which stands for unpaired wise group arithmetic mean technique, in which in this technique we actually take the different means of different distances, and then we combine those differences. And if we go into the concept of UPGMA, it employs a sequential clustering algorithm in which local topological relationships are inferred in order of decreasing similarity, and a phylogenetic tree is built in a stepwise manner. For instance, decreasing similarity means that if there are, for instance, there are one sequence. If we say this is another sequence, this is third sequence, this is fourth sequence, and this is the fifth sequence. So here you can see that it clusters the tree, it generates the tree based on the decreasing similarity. It means that the sequences which are most similar are clustered together, and then in the next step the other sequences which are close together are clustered, and then the rest of the sequence. So one by one, pair by pair, it actually clusters the sequence. For example, here you can see that 1 and 2 are closely related, so it will make a cluster of this like this. For example, we say that, for instance, this is the cluster 1 versus 2, and then we can easily see that 3 and 4 are related. Now they are related, then it will make another like 3 and 4, and then we know that this cluster is actually composite cluster, is actually closely related to this one, so it will make a cluster of this. So it means we will combine it with this, and then when all the clusters are together, then we know that this will be the last cluster. So you will join this tree into. So based on this is actually the concept behind the UPGMA method, or most of the distance-based methods, right? So if we take an example, right? If we say UPGMA example, right? So for instance, let's draw it here that we have 5 different species, A, B, C, D, E, right? And A, B, C, D, and E, right? Here's this, and now we want to make a tree of these sequences. So as we have discussed previously, that when we are making a phylogenetic tree, first of all we have to build a matrix, right? So for making a matrix, and matrix contains the number of nucleotide differences or protein differences between the two sequences. For example, if we are trying to generate a tree using this tree, now first of all we should have a distance between A and B, A and C, A and D, and A and E, right? Similarly, B against C and B against D and all, right? So first of all, this is just an example, so we just follow the example here that the distance between A and B is 22. What actually is the distance? Distance is P equals to N by L, total number of nucleotide differences. Or you can, yeah, right? Next, here A and C 39, okay, A and D is 39, A and E is 41, right? And B and C is 41, 41, 43, and so on. Here C and D 18, C and E 20, and 10, right? So next we just don't have to write again and again because it's already written. This is a 2D matrix, so we don't have to write the differences again. So first of all, what we've discussed here, that based on the decreasing similarity, right? So first of all, the two sequences which are most closely related are first clustered together. So what do you think? What is the minimum minimum distance between two species is in which cell? Like it means that the most closely related species or sequences A, B, C, if it's a individual or it's a species, whatever it is a gene. So minimum distance between A, B, C, D, E is which one? You can just guess here. That's clearly written here, that if you see that D and E has a nucleotide difference or protein difference of 10. So 10 is the minimum number. So what actually the UPGMA does is actually draw another matrix based on based on this matrix, and clusters these two species together. For example, here you will say that D and E are clustered together. So this is one. This is one. So we write it like this, right? And now we have to make the distance, the matrix again after combining these two, and how actually they combine it. If we draw a tree here, you can see that the distance between A and, just open the presentation along the way so that you cannot get confused. So the next matrix would be like this. Example, A, B, C, and D and E are clustered together, right? So we write A, B, C, and D, E. So what will be the matrix? No, how will be the matrix? How will matrix look like now? We'll just see it in a minute's time. So here you can clearly see that already, if you follow if you open the presentation, or you just follow that A and B distance is 22, 39, A and C is 39. So you just write here what 22, 39, and B and C is 41, right? So now we have to calculate the distance between B and DE, right? So B and DE, actually the distance is the distance of distance between B to DE is, if you see that in the previous matrix, you can see that the distance from B to D is 41. The distance from B to E is 43. So the average distance between B to D and E will be the actual distance in this cell. So what is the average here? 41 plus 43, that is what? 42, right? So we will write here 42. And oh, I mixed the previous one. If you look in the previous, then the distance between D and A and D and A and E is 39 plus 41. So 39 plus 41, like I'm saying that if to be more precise, distance between A to D and E. So that will be 39, because the distance of A to D was 39 and distance of A to E was 41. So we divided by 2 and the actual distance is 40. So we write here this one 40, right? So now in this way the new matrix is filled, and we will the last one is missing here. Oh, sorry. Why it's just I always do dirty work. Okay, so C and D is again, that's distance from C to E was D was 18, and distance of C to E was 21. So 18 plus 20, divided by 2, that will be 19, right? So the tree is now D and E are clustered together like this, D and E, and we have a new matrix here. Now we will fill another. Now from decreasing similarity, now we know that what is the closely related species or sequence in this cluster, that is C to DE. Here you can see that C to DE. This is actually 19, which is the minimum number. So now again we will again make a new cluster here, and that cluster will the tree will be like this. If we say that the tree will be C, D, N. Now this is the we have added another cluster into the tree, and the matrix will look like this. I just follow this so that you don't have to worry about in future. So again it will be A, now B, right? Now B and then what? Now B and now we are combining what? C, D and E, right? Is this the way? Yeah. So this will be one. There is no difference here. C, D, E. Right, so you will write here A, B, C, D, E, like this. Right, so this is the matrix. Now what will be the difference here? If you look into the previous section again, then the distance between A to B was 22. So you'll write here what? 22. And this distance between A to 39, A to C was 39, and A to DE was 40. Right. So we will write 39 plus 40 divided by 2. So what actually will be 39 plus 40? If you just to avoid any confusion, I will use the calculator for this most complex equation of my life. Divided by 2, that is 39.5, right? So just we just write here 39.5. Okay. 39.5. So this is the distance from A to DE, and again B, the distance of B to C was 41, and B to DE is 42. Then again 41 plus 42 is actually what? 41.5. That is 41 plus 42 divided by 32, right? So the actual tree is now, that was first it was DE, then C. Now which is the minimum what is the minimum distance? You can clearly see here that A is actually closely related to B. So we just make another matrix here, and A and B, we will combine A and B together. So these here you can clearly see that D, E, C are closely related, and A and B are closely related. A and B are closely related, and the new tree, the new matrix will actually be A, B, C, D, E, right? And here what? A, B and C, D, E, and this will be the most simplest tree. So A, B distance will be now, and so the distance between AB and CDE will be what? The distance is actually, you can see here that the distance of A to CDE is 39.5, and the distance between B to CDE is 41.5. So we will write 39.5 plus 41.5 divided by 2. So if we just want to make a tree, but here we can clearly see that we now have a clear picture of what will be the final tree. So so just to complete everything. 39.5 plus 41.5, right, divided by 2, that is 40.5, right? So in this, here we will just write here this is the final distance. This is the okay, 40.5. So this is the final matrix. So now after having all the clustered together, now we know that C, D, E are clustered together, and the final tree would be, if I just make a neat final tree, so that will be, if I draw it here, okay, if I draw it here, C, D, E, right, with any other color. Now first was D and E were clustered together, then there was an outgroup of C, and then we had a tree like A and B, and now we can join it and insert a root here, and or if you want to insert a root, and this will be the final tree, that D and E are most closely related. Then there is an outgroup of C, and A and B are most closely related, and then we had this final tree. So you can find the different types in the PPT presentation I have shared with you. There are some branch lengths and there are some small ABC written. Just forget about this for a moment and just have a basic understanding of how the UPGMA method is generated using the decreasing similarity of concept, right? So this is very now you can have also an idea that why the maximum parsimony method is more complex or very stringent compared to this, because here it just have to calculate the overall distance, and then based on the closely related species or sequence, step by step it generates a tree. So that's why it's very fast, and it produces a rooted tree with branch length. It also showed the branch length obviously based on the number of changes. And yes, an assumption of the algorithm is that the molecular clock is constant for sequences in the tree. If there is an unequal substitution rate, the tree may be wrong. Like the meaning of this property of UPGMA is that it do not considers different variabilities, like we have discussed one or two of Kimura model, that it says that transversions are less frequent compared to the transitions, but UPGMA does not take into account these kinds of concepts, and just based on the uniform rate of change, that is known as molecular clock, or based on time the uniform rate of change is expected. Based on that assumption it generates the tree. So this actually can get confusing, the tree may get confusing when we are dealing distantly related species, because the substitution rate varies a lot. It is less accurate compared to the other methods, even compared to the other distance-based methods like neighbor joining and other, but it is faster because it do not take different concepts into account, right? Different algorithms like to calculate the variable substitution rate. Whereas the neighbor joining method is comparatively similar. Again, it makes a tree based on the decreasing similarity. For instance, the neighbor joining tree is actually generates an unrooted tree. First it gets all the sequences and assumes that all the sequences are equally related, and then it generates a star-like structure like this. For example, we have a lot of different, now we are just talking about quickly about the neighbor joining method, just to make sure that all of you know what is neighbor joining. Like for example, neighbor joining method, right? So we say neighbor joining method, right? Neighbor joining method is actually it considers all the tree, all the species or sequences that they are equally related. For example, this is one internal. I'll explain you one, 2, 3, 4, 7. Like it first makes a star-like structure of all the sequences that and considers they are equally related, and then based on the distance, just like we discussed in the UPGMA, based on the distance, it actually generates another. Um, it improves the tree. Like for example, it's if we assume that A and B are more closely related than the rest of the species, then it will first make a cluster of A and B, 1 and 2, and then generates an internal branch. Like if it was an internal branch, which is connected to the rest of the trees, which is again in a star-like structure. For example, here there are 1, 2, 3, 4, 5. So we say that 1, 2, 3, 4, 5. So 3, 4, 5, 6, 7. So now we say that A and 2 are A1 and 2 are closely related, and they are connected through an internal branch to the rest, internal branch and node to the rest of the tree. So based now, if in the next step, if there is a if 3 and 4 are closely related, then it will make another cluster of 3 and 4, and then again join that with the internal branch, and so on. This and generates the final tree. So, but it takes into account different concepts related to the variable changes in the evolution, the variable substitution rates. That's why it is comparatively more accurate compared to the UPGMA method, and but again it is faster compared to the character-based method, and it generates an unrooted tree. And as we discussed previously in our first or second lecture, that how to generate an outgroup or to generate a rooted tree, it takes, after generating the unrooted tree and all the clusters, it takes into account the outgroup method to generate the root of the tree, right? And then it is again, as we said, that it is performs well under substitution rates vary among lineages, and thus neighbor joining should find the correct tree if distances are well estimated, right? Thank you very much. Now I think you have a basic understanding about how an UPGMA method works, and if we just revise it, that we based on the matrix, we calculate the closely related species and then generate a tree, and then we regenerate the matrix and then add one by one the one node into the tree, and then we have the final tree when the matrix when there is no taxa left.